Dr. Amit Bhattacharya, Associate Professor at Department of Zoology, Ramjas College, University of Delhi. The title for my presentation for today is Vector Borne Diseases. Now, before going into the vector borne diseases, we will discuss about what exactly vector borne diseases are, what do you mean by vector borne, and how exactly they are transmitted and how the replication and the processing of these vector borne diseases takes place. Now, the vector borne diseases are known for many years over the period of time, and it has infected most of the parts of the world. It has been said that about more than 17% of all the infectious diseases, which causes about more than 7 lakh deaths annually, are vector borne diseases. They can be either caused by parasites, bacteria or viruses. One of the example of vector borne diseases is malaria, dengue, chikungunya. So, we will be discussing some of these interesting and very lethal diseases over a period of time. Now, the malaria causes about estimated 219 million cases globally, and it results for about 4 lakh deaths every year. Most of the deaths occur in children under the age of 5 years. Now, about 3.9 million people in over 129 countries are at risk of contacting dengue, with an estimate of about 96 million symptomatic cases and an estimated 40,000 deaths every year. So, we can see how lethal these diseases are developing over a period of time. Now, there are a number of diseases which have come into the existence, like the chikungunya fever, Zika virus fever, yellow fever, West Nile fever, Japanese encephalitis, tick-borne encephalitis. Now, we come into the topic what exactly vectors means in these particular vector-borne diseases. Vectors are basically the living organisms that can transmit the infectious pathogens between the humans or from an animal to humans. Now, if we see the picture over here, the vectors can be the mosquitoes, sandflies, ticks like this one. So, what happens is they ingest the disease producing microorganism or the pathogen during the blood meal from an infected host, which can be a human or a animal, and later on they transmit these particular pathogens into a new host after the pathogen has replicated. So, that is what the vectors constitute. Now, we come into a very interesting term which has come up, which is the zoonotic diseases. Now, this zoonosis is basically diseases or the infection that are naturally transmissible from the vertebrate animals to the humans. The zoonotic pathogens may be the bacterial, viral, parasitic, or may involve many of the unconventional agents also. So, they are spread to humans through direct contact or through food, water or the environment. So, the zoonotic diseases has been found in number of countries. Now, there are about 200 known types of zoonoses. So, these zoonoses comprises a large number of new and existing diseases in humans, and many of them are like the malaria, dengue, chikungunya, Zika virus, Ebola. So, these are all coming up from the zoonotic diseases, means they are coming up from the animal host systems and they are transmitted into the humans. Now, if we see the diagram over here, how exactly the zoonotic diseases they get into the human transmission systems. Now, there are domesticated and non-domesticated animals which are there. So, the wild zoonotic animals are basically the reservoir for these particular zoonotic diseases. Now, one of the zoonotic diseases which was spread a few years back was the COVID virus. We all have heard about this zoonotic diseases, and we think that this comes from the bats actually. Now, what happens is whenever the humans comes in contact with these kind of wild zoonotic reservoirs or the vectors, there is a chance that they may spill up onto the humans, into the human. Now, that is how the transmission takes place and they start infecting the human host systems. And there are number of reasons why exactly these things can happen, because many a times the habitats are overlapping and there are various other environmental factors due to which the transmission takes place. Now, the zoonotic diseases or the zoonoses have been divided broadly into three major classes. Now, as we just discussed, zoonoses are defined as those diseases or infections which are naturally transmitted between the people and the vertebrate animals. The first class under this zoonotic disease are the endemic zoonoses. Now, these are present in many places and affect many peoples and animals. The next one comes is the epidemic zoonoses, which are sporadic in temporal and spatial distribution. And the third one are the emerging and re-emerging zoonoses, which are newly appearing in the population or have existed previously but are rapidly increasing in incidences and geographical region. So, many of these vector-borne diseases are also a part of these zoonotic diseases, means they have come from the animal system to the human host over here. Now, there are various strategies and directions to control these zoonotic diseases, which includes building up of effective collaboration between the human and the animal health sectors, improving the surveillance for easy and early detection of these diseases threat in humans, and strengthening of the laboratory diagnostic capacities for novel pathogens. So, every years and every month we may hear some new pathogen has been spreading up or it has come up. So, there has to be some laboratory diagnostic capabilities which has to be enhanced so that it can easily detect these novel pathogens which are coming up. Now, the next strategy is the improving of the case management and infection control. Then comes the integrated vector control management, reducing the transmission through social and behavioral intervention, and developing epidemic preparedness and response capacities for emerging zoonoses. So, these are all the strategies, directions which are involved in the control of these zoonotic diseases and play a very integral part of how the control and the treatment regime takes place. Now, if we see the diagram over here, the image over here, this shows the viral emerging diseases. So, over a period of time, if we see, number of viral emerging diseases has come up. Many of them are the zoonotic diseases. For example, the dengue virus, then the SARS virus, then the Ebola, flu, Zika virus, swine flu. So, all these viruses are coming up and emerging over a period of time. Now, the India government has developed a National Center for Vector-Borne Diseases Control, which comes under the Ministry of Health and Family Welfare, which is being maintained by Government of India. So, this is the screenshot of that particular institute's website. So, if we visit this website, we can get the information about all the vector-borne diseases and the zoonotic diseases, as well as we get the information for the treatment regimes, the diagnosis, and what are the measures which can be taken up for prevention and the control of these particular zoonotic or the vector-borne diseases. Now, we come into the overview of some of the important zoonotic diseases, vector-borne diseases over here. Now, few of the major vector-borne diseases are malaria, leishmaniasis, Japanese encephalitis, chikungunya, dengue, and filariasis. So, these are some of the major vector-borne diseases, and in this lecture we will take up some of the important one, which includes the dengue, chikungunya, and the malaria vector-borne diseases. Now, the main vectors which helps in the transmission of these pathogens or the microorganisms or the disease causing agents are the vectors. Now, the vectors can be the mosquitoes over here. So, some of the mosquitoes comes under the class Aedes, which transmit the dengue fever, yellow fever, chikungunya, Anopheles, which transmit the malaria, Culex mosquitoes, which transmit the Japanese encephalitis, lymphatic filariasis, West Nile fever. So, we can see the images of all these mosquitoes and how exactly they transfer these particular diseases. Now, there are certain sandflies also which transmit the leishmaniasis and sandfly fever. Ticks also transmit many of the diseases, which includes the Lyme disease, relapsing fever which is there, tick-borne encephalitis which is there. So, there are number of diseases which can be transmitted through the ticks also. Then comes the bugs, CEC fly, which is an important vector for the transmission of the sleeping sickness or the African trypanosomiasis. Then the fleas comes, which have spread the plague conditions and the other condition. Now, this is the image which shows how exactly there is a continuous conflict between these transmission of these pathogens, vectors and the humans. So, we know that over a period of time, due to the various climate changes and environmental changes, there are often cases where new diseases are coming in, or the new vector-borne or zoonotic diseases are coming in, which were initially found or traced in the animal model systems. But over a period of time, due to various cases like the deforestation, overlapping of the boundaries and these things, these diseases are now getting transferred into the human host systems, and they are affecting the humans, because many a times the humans are becoming less with the immune system. So, they can easily replicate and divide within the body, and they can then subsequently affect the various vital organs of the humans also. Now, we come into the first vector-borne disease, which is the dengue and the severe dengue. Now, dengue is basically a viral infection which is transmitted to the humans through the bite of infected mosquitoes, basically the Aedes mosquitoes. It is said that about half of the world's population is now at the risk of dengue, with estimate of about 100 to 400 million infections occurring every year. Dengue is basically found in the tropical and the subtropical climates worldwide, mostly in the urban and the suburban areas over here. Many of these dengue infections remains asymptomatic, means no symptoms comes in, because the person's immune systems may be very good to fight with these particular, but many a times there are number of symptoms which comes up, or the mild illness comes up. Now, the prevention and the control of the dengue basically depends upon the vector control. So, there is no specific treatment for these dengue or severe dengue cases, and the early detection, there are various rapid diagnostic kits which are available, is very essential for treatment of these particular patients. Now, if we see the image over here, this image shows the countries which are being affected or are at the risk of the dengue. So, this is the data which is released by the World Health Organization. So, we can see the tropical and the subtropical countries are mainly the countries which are under the grasp of dengue virus infection and transmissions, in which India is a major part of these dengue transmissions. Now, dengue is regarded as an emerging or established disease condition, and there are 4 dengue viruses which are there, which are nomenclatured as DENV1, DENV2, DENV3 and DENV4, and most of them are being traced that they have originated in the monkeys or and independently jumped to the humans in Africa or Southeast Asia between 100 to 800 years ago. So, we can see the image over here which shows the dengue virus particles which is present within the tissue specimens over here. So, we can see the black dots which are basically the dengue virus specimens which are present within the tissue specimens collected from the sample of a patient. Now, the dengue hemorrhagic fever, which is the severe case of the dengue, was first documented only in the 1950s during the endemic in the Philippines and the Thailand area. Now, subsequently, the dengue virus structure was deciphered out, and it was found that this is a small single-stranded RNA virus which comprise of 4 distinct serotypes, which we just told you, DENV1, 2, 3, 4, which is present. Now, they are closely related serotypes of the dengue virus, and they belong to the genus Flavivirus and family Flaviviridae family, which is there. Now, the mature particle of the dengue virus is having a spherical structure which is a diameter of 50 nanometer diameter, and it contains number of multiple copies of 3 structural proteins which are there, a host derived membrane bilayer and a single copy of the positive sense single stranded RNA genome which is present in the center part. So, we see the structure of the dengue virus. So, dengue virus, as I said, it is roughly spherical in shape. Inside the virus there is a nucleocapsid which is there, which is made up of viral genome, and the C protein is present. The nucleocapsid is surrounded by a membrane which is called as the viral envelope, which is a lipid bilayer that is taken from the host system. So, we can see the image over here which shows the dengue virus structure. So, outside there is a viral envelope which is there, then the nucleocapsid is there which contains the viral genome and the C proteins which are there, and on the surface of them there are various proteins which are there, E and M proteins which are being nomenclatured. So, embedded in the viral envelope are the E and M proteins that span throughout the lipid bilayer which is covering this particular internal part of the nucleocapsid, and these protein forms a protective outer layer which helps the virus to enter into the human cell. So, there are certain receptors which are present in the human cell which recognizes these particular proteins, and that is how the entry into the human cells takes place. Now, this is one of the vector which is shown over here, Aedes aegypti, which is shown over here. The next vector is the Aedes albopictus, which is shown over here. So, these are the main vector or the mosquitoes which transmit the dengue virus. Now, the Aedes aegypti and the other mosquitoes have got a complex life cycle which is covered in 4 phases, which contains the eggs, larva, pupa and the adult stage. The pupa and the larval stage are mainly found in the water, while the eggs and the adult stage are the terrestrial one. So, we can see all these stages and how exactly these stages develops one after another. Now, the entire life cycle of this particular Aedes aegypti, it lasts for about 8 to 10 days in the room temperature depending upon the feed which it is getting up. So, there are 2 aquatic phases which includes the larva and the pupal stages, and the terrestrial phase which includes the eggs and the adults in the case of the Aedes aegypti life cycle. Now, this is a diagram which is showing the primary infection of the dengue virus, how exactly the virus enters into the host system, and it uses the host system to replicate and reform itself so that the other number copies of this particular virus is made within the host cells. Now, some of the symptoms of dengue includes the mild or no symptoms, and they generally comes up after 1 or 2 weeks only. The dengue can be severe, rarely the dengue can be severe or lead to death. If the symptoms occur, they usually begin within 4 to 10 days after the infection and last for 2 to 7 days actually. Now, some of the basic symptoms includes high fever, severe headache, pain behind the eyes, muscle and joint pains, nausea, vomiting, swollen glands and rashes. Some of the severe dengue cases, the symptoms includes the severe abdominal pain, persistent vomiting, rapid breathing, bleeding gums or nose, fatigueness, restlessness, blood in vomit or stool, and pale and cold skin and feeling weak. So, these are some of the severe cases when the dengue is not treated or controlled at the initial level, it may lead to these kind of cases. Now, this is a diagrammatic representation how the virus affects and how the symptoms comes up in the different parts of the body, which includes the brains, the gastrointestinal tract, the bone marrows many a times also get affected, the blood circulation gets affected over here. Now, the dengue severe cases can lead to condition which is called as the dengue hemorrhagic fever or dengue shock syndrome. Now, these conditions are generally severe and there is a risk of mortality which is very high in these particular cases. Now, if we see the diagram over here, this diagram is showing the viruses which are impregnated into the tissue sections over there. Now, we come into the diagnostic, how exactly the dengue infection is treated or it is being traced that whether a person is suffering from this infection or not. So, it can be treated either by direct reactions, which includes the detection of the dengue viral genome, which can be detected with the help of the reverse transcription polymerase chain reaction or RT-PCR, which is done with the help of the blood specimens which are collected from the sample of the patient after 5th day of the illness. Some of the other detection techniques include the detection of the non-structural protein NS1 protein, which is present on the surface of the dengue virus. So these generally start coming up, and if they are detected with the help of the ELISA plates and these things, they also gives us the information that the person might be suffering from the dengue virus infection. Now, there are various serological diagnostic techniques which are also present, which includes the detection of the IgM antibodies specific for the dengue or IgG antibodies in the titer or the circulation. So these are also detected in the serum specimen after 5th to 6th day of the illness, or the detection of the IgG antibody also takes place. So these comes under the serological diagnostic techniques over there. Now the dengue virus genome are generally detected mainly by the RT-PCR, or it can be detected with the help of the ELISA techniques, which generally detect the non-structural protein NS1 dengue antigen, which start coming up 4th day post onset of the symptoms, and it depends upon the specific sensitivity of this assay, depends upon the serotype which is coming up. Now this diagram shows up what are the direct methods which are there for the detection, which includes the virus isolation, genome detection, or the antigen detection. So the specificity of these direct methods are very high, while in the case of the indirect method, which includes the serological IgM, serology IgG collection, the sensitivity is not very high, but there are better chances that it will be easily detected. Now this is a tabular representation which shows the comparison of various laboratory diagnostic techniques which are there, which includes the direct method as well as the indirect method, like the IgG or the IgM detection through the ELISA techniques. Now the dengue serological test generally includes the MAC ELISA, which is IgM antibody capture enzyme linked immunosorbent assay. So these methods are being used. Now there are various ELISA plates which are available in the market. So it is easily available and they can be easily detected. So some of the methods are shown over here. So this is a ELISA plate which is used for the NS1 detection for the dengue over here. So we can see how exactly the blood samples are taken in. Then the serum, from the serum sample, the aliquot is poured onto these micro titer plates, and subsequently the various chemicals are added, and we get a bands which shows whether the result is positive or negative. So these techniques have revolutionized the detection and the diagnosis of dengue infection. Now there are various methods for the control of these growth of these mosquitoes in the surrounding. So we all know about these things. Generally they start growing up in the stagnated water. So wherever there is a stagnated water, it should be released up from that particular place, so that there is no stagnated water, which helps in the growth of these mosquitoes. So I come to the last slide in this part of the presentation, which is the prevention and the control measures. So there are various prevention measures which includes the removal of the stagnated water in the surrounding wherever they are present, the use of the mosquito nets. Nowadays the insecticide impregnated mosquito nets have come up, so they are a very good source of the control measures. So with this note, I come to the end of the first part of the lecture on the vector borne diseases, where we discussed about the vector borne diseases and the dengue viruses. In the next lecture, we will be talking about the chikungunya and the malaria infections. Thank you very much.